Welcome to our Q&A session. I hope you enjoyed our presentation. After Eric concluded his presentation, we held a brief Q&A with members of our science team, including Dr. Earl May, our Senior Director of Discovery Technologies, Susan Cornell-Kennan, Senior R&D Scientist, Matthew Hakar, Research Associate 2, Treyer Wilson, Research Associate, and myself, Jack Clark, Product Manager. We would love to take your questions in real time, but obviously this is a recording, so we can't do that. So please write down any questions or thoughts you may have and email us directly at hello@assayquant.com. We look forward to hearing from you. That was great. Thank you so much, Eric. Yeah, a phenomenal overview of who we are and what we can do. So Susan, I'm just going to pass it right off to you so you can begin our Q&A section. And you are on mute. Thank you. You're still on mute.
Could you speak to why continuous real-time kinetic data is superior to data generated in traditional endpoint assays? And what information can you miss with an endpoint format?
Sure, that's a great question. So I think, with the continuous format, you get much more information that can be both about the target biology and the drug. And so with the target biology, we are routinely seeing, for example, kinases from different vendors. Some will have a lag, some won't have a lag. We can actually do a pre-activation and get rid of the lag, right? If you just did a 30-minute time point or an hour time point, you would not see that. Likewise, we will see different products from different vendors where we will see premature instability. And so after even 30 minutes, the enzyme is dying or dead. And so all of this enzymology comes forward. And there are many other key areas like residence time where you're just doing the jump dilution and you allow it to then recover over time. So doing that with time points is very difficult. With us, you just set it up and then just watch it over time. Likewise, we can see time-dependent inhibition, so the different shapes of the progress curve with different drugs. A lot of interest in time-dependent inhibition now, and we see it right away in the forward progress curves, and then we can do appropriate follow-up to actually study it in greater detail.
Okay, thank you. I have a follow-up question. What other advantages makes the PhosphoSense platform so enabling?
Sure. So I think that the continuous format is really dramatic in terms of liberating your staff so that they're not spending time doing experiments all day long if they were doing time points. The ease of use of the assay is really incredible, that it's add and read and you walk away and do something else. We are also optimizing various automated data analysis templates to make data analysis that much more automated as well. And so the whole net effect is that it really gives you the right information with less effort so that you can get at the critical differences in your lead compounds so that you're bringing the best compounds forward to the clinic.
Thank you.
Perfect. Earl, how about you ask a question?
Sure, that was great, Eric. Can you share with us why you would do kinome profiling with ATP at Km when you could run it at physiological concentrations like 1 millimolar?
Yes, yeah, it's interesting that, you sort of, if you look across all of our clients that are doing profiling with us, I mean, some are emphatic. We want millimolar concentrations. We want a cell correlate. We really want to know how this compound is going to behave relative to our cell-based assays and ultimately in the clinic in terms of specificity, right? Others are, no. We want to look at the ATP-Km. We want to know the maximum amount of inhibition we see across the complete kinome. And, the answer is that you want to do both, right? And we can do both with our format. And so that's really what's powerful. There's so many profiling formats out there where it's really based on P32 ATP and 10 micromolar ATP. So they can't go to millimolar, right? We can do either, right? And we can even do it with the same run to look at both concentrations. So the key thing is getting more information then with your drug so that you really have greater confidence in terms of what you're bringing forward, either in terms of SAR or your final leads.
Yeah, that was a great answer, Eric. Treyer, I'm going to hand the virtual microphone over to you.
Thank you, Jack. So I have a couple questions. So I know our clients work with a wide range of compounds. And my first question is, with covalent inhibitors, what is the best way to analyze these compounds? And what could you be missing if you only reported IC50s?
Yeah, that's a great question. So you see that routinely. You look in the literature at covalent compounds and they're doing IC50s. And the problem is that IC50 is changing over the course of the reaction. So you can even do pre-incubations, but what you really want to see is the time course so that you get the initial reaction rate, you get the K-ops, you get the final reaction rate. And with that global analysis, that global fit, you then get very accurate assessment of the potency of the compound in the terms of a Kinact over Ki value. And that's how you want to report, because then it's independent of the time point and the method. And that's what our platform really enables, because those are complex experiments. And you know, we run a 24-point titration of the inhibitor to give us that data. It's very powerful, and we're seeing many groups then use that to bring the right compounds forward to the clinic.
Awesome. And then for my final question, some compounds exhibit autofluorescence and can have interference. How does the PhosphoSense platform address this concern?
Yeah, that's also a good question. So you still see a lot of compounds that have high autofluorescence, and especially in the beginning stages when we're running 10, 20, even 50 micromolar compound, because they're initial, leads and they want to optimize it further, but you're seeing a lot of autofluorescence. And so with the kinetic format, because the background won't change, right, you can just subtract that autofluorescence from your total progress curve to get the corrected. But if the autofluorescence is really high, you can't do that. And that's where we just switch to the PhosphoSense red format. And so using europium with the time resolved fluorescence, 100 microsecond delay is long enough to allow any compound autofluorescence to go away, right? Those are nanosecond time scales. And so when we're looking at 100 microseconds, we're only seeing the signal from the SOX fluorophore with europium.
Thank you.
Yes, great.
Awesome. Yeah, that was a great question. And then Matthew, I'll let you have the last question.
Thanks for the presentation, Dr. Schaefer. I just have a few questions for you. So say I'm looking for an assay that isn't commercially available. Do you have a process for developing new assays?
Yeah, so we've worked a lot on this. So as highlighted in the slides, this is a common request. So it's either that there isn't a good assay for that target, right? There may be assays on the market, but they're not robust assays that really allow you to go down to very potent compound concentrations. So groups will also say we're using a protein substrate, and we don't want to use that, for all of our lead optimization assays. Other groups will come to us and say, we have an assay, but it's bottomed out. That means that we're having to use enzyme concentrations that are, at least twofold higher than the IC50. Can you do anything for us? And for all of those scenarios, we can rapidly turn around a very robust assay using this milestone development process, where it can result in 100-fold increase in the reaction rate, which gives you, almost a comparable increase in sensitivity. So you're going from using micromolar or high, nanomolar levels down to single-digit nanomolar. So very dramatic.
Yeah, that's great. And my second question is, are all the sensor peptide substrates generic, or is it possible to identify sequences that are selective for the target of interest? And if we can develop more selective substrates, could they then be used with crude cell or tissue lysates?
Yeah, that's perfect, right? So I think initially, we had a lot of people that we worked with just as collaborators, and their initial comment was, you're not going to get selective peptide substrates. And the answer is no, we absolutely can. And that's the power of using high throughput peptide synthesis and being able to use the PhosphoSense platform as a screening tool, because you just see it on the screen immediately, right? And so it's very easy to find the needles in the haystack. And what we find when we systematically change, a given position with 16 different amino acids is that it can have a dramatic effect in terms of selectivity, in terms of reaction rates, in terms of signal to background. And so we can harness all of those readouts to get to a much more robust assay that also now is really selective. And we don't have crystal structures yet, but we really believe that it is presentation of the peptide in a way that mirrors the native substrate. And again, we see 100-fold change in the reaction rate. So that's what we think we're doing. And it can be a minimal change actually in a 15 or 20 amino acid peptide sensor peptide sequence that results in that increase in performance. So we want the minimum change in a physiological sequence that gives you that better performance.
Yeah, it's good that you have that flexibility.
Yeah, and I think taking that on, like I said, to the crude cell or tissue lysates allows us to get at the complexes and ask how are individual clients' drugs working with those more physiologically relevant complexes. Because ultimately that's what's going to happen in the cell and in patients.
Awesome. Yeah, great question, Matt, and thank you, Eric, for answering it. So I do have one more question, and that's, where do you see AssayQuant in 2 years?
Well, we've grown a lot, right? So we've gone from 5 people to 25 people in the last several months. And we really see AssayQuant's products and services continuing to grow because we're providing an unmet need. And the whole field is really alive right now with different chemistries. We have covalent inhibitors that are reversible. We have reversible inhibitors that have really long residence times. We have, just a plethora of different phenotypes of these compounds that we're able to harness. And our clients are then able to apply those benefits in terms of compound performance in the clinic. And so we see individual companies going through this process when they're working with us much faster. They're getting fast track status through the FDA. They're getting best in class status for their compounds. And we take great pride in enabling that. And we're excited, our clients are excited. And we think that one offshoot per Matthew's questions about why do you want selective sensors, if ultimately we can actually get to the point of offering diagnostic tests that would allow you to understand which patients are going to respond to a drug, which patients, once they receive the drug, what is the change that occurs in their signaling pathways, in their cells, in their tumor, if it's a cancer, right, that we need to detect so you know when to change. And if you change and you do, for example, develop resistance, right, you need to know when to change. And you need to know what drug regimen to provide so that the patient then sees much better treatment regimens. And I think that's the number one opportunity, that if a drug gives a patient who has cancer 6 months, it's like, why couldn't that be 6 years? And that's what we want to deliver. We want to help our clients really come up with better treatment strategies and better drugs that deliver that efficacy.
That's awesome. A super inspirational message. So I think we should end it there. So thank you so much for Eric for your time today and our expert panelists. And for anybody in the audience who's interested in learning more about AssayQuant and how we can enable your drug discovery and development, or if you had any questions during Eric's presentation, please reach out to us at hello@assayquant.com. We'd love to learn more about your efforts and answer any questions that you may have. So thank you very much and have a great day, everybody. Thank you so much for your time today, Eric and team. For anyone who is interested in learning more about AssayQuant and how we can enable your drug discovery and development, or had any questions during the presentation, please reach out to us directly at hello@assayquant.com. We would love to learn more about you. We look forward to hearing from you. Have a great day.